Deregulating cellular energetics, a hallmark of cancer. Cellular energetics obviously involves cellular metabolism. Taking a look at normal glycolysis, we can recall that one glucose molecule can be processed to generate two molecules of ATP, 2-pyruvate, and it reduces 2 NAD plus to NADH. The NADH are subsequently shuttled into the mitochondria to participate in oxidative phosphorylation, where they can be oxidized and reused in more glycolytic reactions. Pyruvate is converted into acetyl-CoA for use in the TCA cycle. This all occurs under aerobic conditions. Under anaerobic conditions, the end products of glycolysis remain the same, but NAD plus needs to be replenished through fermentation by lactate dehydrogenase. Tumor cells produce a lot of lactic acid, even under aerobic conditions. This was first described by Otto Warburg in 1926 and termed the aerobic glycolysis, or the Warburg effect as it's become known. It describes the preferential selection of using glycolysis over oxidative phosphorylation and the TCA cycle, and thus an overproduction of lactic acid even under aerobic conditions. Warburg identified this by directly measuring the amount of lactic acid present in the blood, both on the arterial and venal side, to note a difference. Table 3 shows the lactic acid present before and after different normal tissues. As you can see, they are approximately equivalent. Table 4 shows the lactic acid present over 10 observations from a particular tumor, the venal side being markedly higher than the concentrations of lactic acid in the arterial side. These two here appear to be noted as outliers. Table 5 shows the amount of lactic acid pooled within a cavity bored into a tumor, showing that there is a significant amount of lactic acid generated by the tumor. So, why ferment pyruvate into lactic acid? Is there something wrong with oxidative phosphorylation in tumor cells, the TCA cycle, the mitochondria? Are things not getting shuttled in? Somehow, normal metabolism is getting disrupted around the last stage of glycolysis. So, one experiment to determine the expression of glycolytic enzymes uses bioinformatics to query the National Cancer Institute's Cancer Genome Anatomy project and takes a look at the expression. They looked at 24 different cancers out of a total of 49 available, but due to what seemed to be a lack of data in 25 of them, they only looked at the 24. What they found was an upregulation of nearly all of the glycolytic enzymes, including the last step, pyruvate kinase. Pyruvate kinase is known to have 4 isoforms in mammals. An L and R form, which are found in liver and red blood cells, is located on chromosome 1. An M form, which is then also divided into M1 and M2, is located on chromosome 15. The difference between M1 and M2 is that they are splice variants. M1 includes exon 9 and not exon 10, and is expressed in almost all adult tissues. M2 includes exon 10 and not exon 9, and it is expressed in embryonic cells. Analysis of pyruvate kinase present in tumor cells shows that it too is protein kinase M2. Since M1 and M2 are splicing variants, there is likely some sort of RNA binding protein which is impacting the splicing decision. So, radioactively tagged oligoRNAs were created to cover the splicing sites of exons 9 and 10. They were mixed with nuclear extracts of helicels, ultraviolet cross-linked to preserve the interaction, and then run on SDS page. What we observe is a radioactive signature around 37 kilodaltons. So clearly there is something interacting. We can then run RNA affinity chromatography with Kumasi staining to visualize the proteins binding around the same size. You can then extract the bands that are occurring around this area, and then you can identify them with mass spectrophotometry. These bands were identified to be 3 heterogeneous nuclear ribonucleoproteins that are known splicing repressors. So, to test how these impact pyruvate kinase M1 and 2 expression, these can be knocked down. Looking at the gel, in the control, we can see that M1 is hardly, if at all, expressed, whereas M2 is expressed to a great deal. Knocking down and interfering with the varying iterations of the 3 splicing suppressors, we can see that M1 is increasingly expressed, but M2 is increasingly downregulated. The same thing happens when we look at a large population of cells as well. So, c-Myc is a known oncogenic transcription factor and was suspected of being involved in the regulation of these splicing repressors. So, c-Myc was also inhibited, and the expression levels of the splicing suppressors were reduced, as we can see compared to the control. Similarly, M1 was being produced more, and M2 was downregulated a bit. Looking at how PKM2 expression affects tumor cells, knocking down PKM2 expression with RNAi reduces cell proliferation, and transfecting a normal PKM1 restores the cell to a normal growth pattern. If you noticed on the earlier slide, there were 2 bands labeled as M2. That's because M2 has 2 forms as well, a tetrameric form and a dimeric form. The tetramer has a high affinity for phosphoenol pyruvate and generates ATP. The dimer is mostly inert. PKM2 also has a multitude of additional interactions. The glycolytic intermediate fructose 1,6-bisphosphate is an allosteric activator which promotes dimerization of the dimer into the tetramer. An intermediate from the purine biosynthesis pathway also promotes normal pyruvate kinase activity when the cell is starved for glucose. Post-translational modification of protein kinase M2 can block tetramerization, and ERK2 can phosphorylate PKM2 in such a way that it can be imported into the nucleus, where it then serves to upregulate the expression of c-Myc, which, if you see it coming, upregulates the expression of GLUT1, which allows more glucose to be taken in by the cell, lactate dehydrogenase A, allowing more replenishment of NAD+ and producing more lactate, and the splicing suppressors that promote PKM2, creating a positive feedback loop. So how does the PKM2 get into the nucleus? So we can see that under EGF present and not, we can see the immunofluorescence of the cells with the darker centers of the nuclei. And if we flag PKM2 versus PKM1, we can see that in the presence of EGF, PKM2 is being expressed throughout the cell, including inside the nucleus, whereas PKM1 is not. Similarly, we can test to see where ERK is binding. In the presence of EGF, we can co-immunoprecipitate both flagged PKM2 and ERK. And we can see that only with the PKM2 is ERK binding to it. So, what exactly is ERK doing in order to get PKM2 into the cell? Well, Using a kinase assay, we can explore how ERK2 affects PKM2. Looking at the sequence information, we can see that PKM2 has an ERK-specific pattern around serine 37. We can develop a antibody specific to target this serine 37, and we can see that we can get it back on these preparations. We can also see that ERK2 is phosphorylating the PKM2, and we can also see that at serine 37, when we do a site-directed mutation and change serine to alanine, we do not get phosphorylation there. So, checking the site-directed mutation versus an phosphorylation analog, we can see that the mutated version of PKM2, similarly to PKM1, does not enter the nucleus even in the presence of EGF. However, the phosphorylation analog can enter the nucleus despite the lack of having EGF to stimulate ERK2 to phosphorylate PKM2. So, why does any of this matter? Well, when you stop the last step of glycolysis, you're not stopping any of the steps above it. This changes the flux of the system, making moving intermediates down the pentose phosphate pathway to produce nucleotide and amino acids very favorable. These are absolutely critical for a cell rapidly dividing. Does anybody have any questions?